Plutôt faire ta, mais heureusement chez la droso c'est assez simple. Il y a des animaux chez lesquels c'est plus compliqué.
Okay, good morning everyone. Welcome to the seminar. So as usual, we are on the webinar mode. So people online, do not hesitate to ask your question in the Q&A tab and to report any technical issue in the chat. And with that, we're going to start. So thank you very much, Simon, for introducing today's speaker.
Hi everyone. So this is Caroline Fabre, the great-great-great-granddaughter of Jean-Henri Fabre, the famous entomologist, and as well of what's the name of the other Fabre who has a museum in Montpellier. So you're from the family of one of these two.
I feel at home here.
Well, that's great. So Caroline did a PhD in Cambridge, working with Peter Lawrence, where she was working on the neurodevelopmental aspects of Drosophila, which is very obscure to me. But I guess they have neurons. Then she moved to Oxford, and then that's where she discovered that Drosophila use those types of vibrations to communicate. And she went back to Cambridge, and that's where she started also working on the pet species, but I don't know if you're going to talk about this much today. And since about a year, she's now in France, in Versailles, and with money from Europe and the region. And you may have met Caroline before if you were in Montpellier in 2010, because she was playing the trumpet in La Fête des Fanfares. So, appreciate that.
Well, thank you for the intro. Hello, everybody. Can you hear me at the back? Yeah? So it's very nice to have the opportunity to present to you what we've been doing in the Drosophila Bioelectrochemology group. So thanks to Simon and the SEM committee for the invite. And the work I'm going to show you today was done at Cambridge, where I was before, until I moved to Paris last year. And so I don't know if some of you, not all of you might know that, but we're discovering more and more that many animals are actually sensing vibration in the ground, and they can use these informations to these vibrations to gain information and to communicate with one another. So this is called seismic communication or substrate-borne communication. And the information they can gather in the ground can be very important for the survival of the animal and the survival of its population. And so I thought I would show you a very striking example of animal sensing vibration in the ground. Oops. And this is in 2004, during the tsunami that hit Asia. And so during this, after this tsunami, there was a tragic number of human casualties. But what was surprising and unexplained at the time was that the number of non-human casualties was actually very low. And this is because most of the animals actually left the area before the deadly wall of water hit it. And so this led to the headlines you can see in this page here, with some people even wondering whether animals had an extra special sense to sense disasters ahead. But researchers know that they didn't have an extra special sense. They just used their highly sophisticated sense of touch, and they sensed very early on that some very bad vibrations were coming towards them, so they left the site before the waves arrived. And this sense of touch to vibrations is often overlooked, but it's actually very useful to us humans too. So we use it, for example, when we manipulate objects. It helps us detect the texture of this object and the frictional properties of the object we're manipulating. And maybe the most striking example for us humans using our sense of touch to vibrations is when we are able to read the braille language with the tip of our fingers. So the vibration acuity in the tip of our fingers is very high because we've got vibration sensing receptors in high number localized there. And so basically when we move the tip of our fingers on this special paper with raised trims, it triggers vibrations on our fingers, which we're able to discriminate. So as I said, in the case of animals, the sensing vibrations can be very important for their survival and for the survival of the population. And this is the case of the very varied examples I'm showing you here. And we believe as well hundreds of thousands of others which are not all identified yet. And so I'm showing you here the elephants. So elephants are able to sense vibration in the ground. And what happens is that they produce vocalizations with their trunks. And these vocalizations couple to the ground. And they can travel for miles to other family members. And they use these signals to gather family members over large distances. And if you watch documentaries on elephants, you might notice that they often have the front of their legs or the tip of their trunks on the ground. And this is because it's been shown that these areas of their body are very rich in vibration sensors. Here you can see the fiddler crab, and it's been shown that the male fiddler crabs can tremulate its large claw very quickly, and this generates vibrations in the sun, and this attracts a female to its burrow. And in the case of the red-eyed tree frogs, the male tremulates the entire body on the plant. These generate vibrations on the plant, and these are recognized as aggressive signals for another male who is not going to go on that plant. So there really is a silent vibratory world out there, which we are discovering more and more. But it's been a challenge to study this species. As I said to you, I mean, it's difficult to record the vibrations because we can't hear them, we can't sense them. And also most of these animals are not easy to study in the lab, and are not genetically tractable. However, some years ago, I realized actually that seismic communication also occurs in Drosophila melanogaster, which you will know is a very easily genetically tractable model species. So how does seismic communication look like in flies? So I thought I would first show you an example of courtship, which is where I realized this type of communication was happening. So here you can see a male following a female, and it's a iconic model system to study in a series of interactive behaviors, and it's been shown that both individuals use a combination of visual, olfactory, gustatory, and acoustical cues during courtship. So I hope you can see here the male, for example, extending his proboscis here, trying to touch the female and gather chemical cues. You can see him extend one of his wings, and he's vibrating it very quickly, producing a song, which we call the love song, the serenade, which is species-specific and is very important has been shown to be very important for female receptivity. And at some point, the female gather these cues. They gather cues from one another, but the female gather the cues from the male, and she's gonna become receptive and become immobile. And this immobility will allow the male to attempt copulation and for reproduction to happen. So a key in a successful courtship really is for the female to at some point become immobile, so copulation and reproduction can happen. And when I started looking at many pairs of courting flies and looking very carefully at this behavior, I realized that actually, it wasn't clear which of these cues of the male were really responsible for female immobility, whether it was the love song or something else. And when I started to look at these movies, I realized actually when the male performed the love song with his wing, often the female was actually walking away. She wasn't really displaying immobility. However, I noticed this behavior that the male was performing and wasn't described in the literature, where I hope you can see is performing this tiny up and down movement of the abdomen. And when he was doing those movements, I noticed that the female was immobile. So I'll show it again, and you'll see that sometimes she's immobile while grooming, and sometimes she seems very attentive to the male. So you see here, he's tremulating his abdomen, and she's grooming, but she's not moving. And now he's carrying on doing it, and she seems very attentive to him. So this is the abdominal up and down movement, which I call abdominal tremulation, at high speed and higher resolution. And so this led me to wonder whether this could be a signal to trigger female immobility. And I don't have time to show you everything today, and this is published if you're interested, but I showed that these abdominal movements were not signaling via chemical cues, nor acoustical cues, nor visual cues. So at this point, I was left with just one hypothesis left to test, which was whether this tremulation could be generating substrate-borne signals in the ground. And as I said to you, these substrate-borne signals are quite difficult for us to identify and record because we can't. They're not obvious to us. We need sophisticated tools, especially when you're dealing with tiny flies like Drosophila. And so I had to use a laser Doppler vibrometer. And the way it works is that you target the beam of the laser onto the substrate on which the flies are courting. And this allows you to identify tiny variations in the vibration pattern of the substrate. And so when I did that, indeed, I observed that the tremulation were generating vibration in the substrate. So there are very regular pulses in the substrate, about 5 hertz, so 5 vibrations per second. And I was hoping so we can digitize these signals. So they are not acoustic signals, right? They're in the ground. But we can digitize and transform them in audible frequencies for us to have a feel for them, really. But I'm not sure it's going to work because we were not really able to. Maybe some people in the room. Otherwise, I'm going to mimic them, and they're like ta 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 ta. They're a bit boring in Drosophila. They're more interesting as other insects, but it's easier for me to mimic. And then again, this is published, so I won't have time to show you, but using monitoring simultaneously the behavior of male and female in many courtship movies, and using special bespoke algorithms as well as genetic and surgical manipulation, we were able to show that these vibrations indeed coincide and promote female immobility during courtship. And so the next question we had for us Drosophilists who know that flies actually court in very soggy substrate was whether this vibration can actually transfer in such rotten material, right? So we decided to test the most obvious substrate in which we know Drosophila court and feed, which was sort of rotten banana, apples, and also the fruit of the prickly pear cactus, because there are some reports suggesting that this is a wild natural substrate for Drosophila. And so that's what we undertook to do with Eleanor McKelvey. And again, we use laser vibrometry and special softwares to analyze the vibrations. And what I'm showing you in this slide is in here, in the x-axis, the frequencies that make up the vibrations we are recording, and in the y-axis, the amplitude of each of these frequencies in the signal. And what you can see very quickly is that we were able to record signals in all the substrates we tested, so like banana, prickly pear cactus, and apple. But you can see that there is modification and filtering of the frequencies that make up the signal depending on the substrate properties in which we are recording. So for example, the signal recording was really good in the prickly pear cactus and not so loud on the apple. And to compare, this is a very reflective foil membrane that's very good at propagating the vibration, which we use as a comparison. One thing we noticed was that the frequencies between 200 and 400 Hz were always present in the various fruits we tested. But basically, there's quite a lot of variations. And I was hoping to show you the difference of amplitude. Maybe you will hear the cactus, because it's quite loud. So the signal was quite good on cactus fruits. But now, how does the female respond on these different substrates, where we seem to have variation in the properties of the signals? Well, actually, the female responds very well on all of these substrates. So in this figure, what I'm showing you is each dot is a pair of fly that we recorded. And for each pair on each substrate, I'm showing you the proportion of time the female is spending immobile during the tremulation. So we are just taking the time the male is tremulating, and we are looking whether the female is immobile or not. And you can see that on apple, banana, and cactus fruits, despite the difference we saw, the female shows very high level of immobility response to the tremulation, in comparison to when we put the pairs on a foam substrate that was very bad at propagating the vibrations, where the response, the immobility response of the female was really poor. So a few conclusions here. We saw that there is dampening of the signal, the vibratory signal on substrate, more or less depending on the substrate properties. It is possible that the original tuning of this vibration was for the cactus fruits. So we've got this concept of tuning in substrate-bound signaling field, where we think animals choose the plant or the substrate on which they're going to produce these vibrations. So you saw it was really good on the cactus, but the signal, the response of the female was pretty good on the other substrate too. And we believe this is because in the case of flies, it's a short-range signal. The flies are very close to one another when they're courting. It's not like the elephant, which has to propagate its signals for miles. And so probably what the female is getting is good enough for her to respond. We're still not sure what is the meaningful information for the female in these vibrations. Maybe it's this 200, 400 range, which we found in all substrate recordings, or maybe it's more what I showed you earlier, the ta-ta-ta-ta-ta, the repetition rate of the signals. So then the next question we wanted to tackle was, how do the female receive these vibratory signals in the substrate? And obviously, for us, the first place to start was to look at the female legs, because the female's legs are directly in contact with the substrate. And again, with Eleanor, we undertook to do this. And the way we did that in flies is that we used a collection of Driver-Galfor lines, which drive expression in subset of neurons in the legs. And we're able to combine those lines with effectors that can disturb the activity of the neurons, either inhibit the activity of the neurons or activate their activity. And we can also use RNA interference to knock down the expression of specific genes in this subset of neurons. And we can use fluorescent markers to visualize these neurons that we're playing with. And so we've got engineered females, which are pairing with normal males. And we observe their courtship, and we're looking whether in these females where some of the neurons are not functioning quite normally, what is their response to the substrate-bound vibrations? Do they still remain immobile? Do they stay immobile less? That's what we're looking. So this is one of these experiments that I'm showing you in this slide. And here we disturb the activity of two subsets of neurons localized in the femoral chordotonal organ of the leg. So this is the femur of a female leg, and this is the femoral chordotonal organ. And you can see that when we knock down the activity of the A neurons here during courtship, the female response to the tremulation is much lower on average than control. Again, it's the same as earlier, right? Each dot is a pair, and I'm showing you the proportion of immobility response of the female to the tremulation. So you can see it's much lower when we knock down the activity of these A neurons, which is not the case, for example, when we knock down the activity of these other B neurons here, where the female immobility response is as high as controls. So these A neurons seems to be important for female to respond to the vibrations. And what we next did was to artificially activate these A neurons constantly during courtship. And what we noticed is that when we did so, the female were significantly more immobile, even when we were not, even when the male was not tremulating. As if the female thinks she's getting information, she's getting vibratory information, so she's staying immobile. So now we wonder how do this A neuron respond to the vibrations. So substrate-bound vibrations are a type of mechanical stimuli. So we expect the flies to use mechanosensation to sense them. And mechanosensation usually functions through mechanically gated ion channels, which are ion channels that are activated by the deformation of the neuronal membrane, which leads to the passage of ions through the channel, and then to action potential in this neuron and activation of the neurons. And so we tested three mechanically gated ion channels, which we found were expressed in the A neurons, non-Shug and TRP gamma, which are part of the large TRP family, which is conserved all the way to vertebrate, and Piezo, which is a large transmembrane ion channel, which you might have heard about because it was the subject of the Nobel Prize in 2021, and it's very important for humans, vertebrate and human sense of touch as well. And so when we depleted these three ion channels in the A neurons, we noticed that when we did so with non-Shug and Piezo, we obtained a significant decrease of the female immobility response to the tremulation. But this was not the case, for example, when we depleted TRP gamma in the A neurons. So non-Shug and Piezo seem to be important to mediate the vibration response of the female in the A neurons. And this is quite important for reproduction, because what we observed was that when we depleted non-Shug and Piezo in these females, in these A neurons of the female, reproduction was decreased. So copulation was much less likely to happen because the female is not so receptive to the male. And so these pairs basically are not as likely to reproduce as control pairs. So this is where we are now with this model where we think the femoral chordotonal A neurons are important to receive the vibration in the female leg. And this is mediated by non-Shug and Piezo. And we think that this is this role of the A neuron is quite important for fly reproduction. So I also wanted to show you what we've done with other species, because we've realized that this seismic communication is not just for Drosophila melanogaster, where I first discovered it, but actually we've now recorded it in many other Drosophila species from these different groups. And we've noticed, I'm showing you some examples here, we've noticed that the repetition rate that I mentioned to you could maybe be informative for the female, varies from one species to another. And here you can see some species I'm showing you which are tremulating quicker than Drosophila melanogaster. And for example, Drosophila suzuki tremulates more than 2 times quicker than melanogaster. Okay, and I wanted to focus more on this one species that I really like, Drosophila persimilis. And when we observed its courtship, we noticed that it has this amazing display where the male here is, oops. The male is displaying his wings, it's rising its abdomen upward and tremulating, and it's feeding the female a nuptial droplet. So I hope you can see it's vibrating the abdomen. And so we decided to look at this species for the reason of and record it on the laser. And so here, this is actually the beam of the laser. The male is here and the female is here. And I'm just showing you the boot of tremulation. Okay, these are bigger, so they're louder. So we realized that actually these males were producing tremulation, substrate-bound vibration while raising his abdomen upward. And this was interesting for us at the time because at the time wasn't clear whether actually tremulation were produced by striking the substrate. And this species clearly shows that you don't actually need to touch the substrate to make this tremulation. They're actually produced by impulse, so probably by pressure on the ground. And this is also a nice example of the versatility of substrate-bound signaling, where here it's integrated in this really complex display posture and feeding behavior. And again, there's clearly a species-specific aspects to the interpulse interval. I also wanted to show you and the other species I mentioned, Drosophila suzuki. So you might have heard, there was some people working on it here, that Drosophila suzuki is creating huge waste and economical damage in the fruit industry because it's laying its eggs in fresh fruits instead of rotten fruits like Drosophila melanogaster. And it's now considered the main economically devastating species in the fruit industry. And it's pretty much everywhere in the world. I wanted to show you how it sounds, but I don't know if you're gonna hear it. Wow. So it's been much quicker than melanogaster. So it's 2.5 times quicker. So ta 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 ta. I don't even know how they managed to do these vibrations. But what we notice is that they spend much more time than melanogaster interacting with this substrate-bound vibration. They spend almost half of their courtship time doing so, and it's only 30% of the time for melanogaster. So we think they're relying quite a lot on this mode of communication. So we got interested to know if we could maybe disturb this and disturb their courtship and their reproduction. And we've recently, with Stuart Russell, been able to find a treatment that is able to decrease their egg laying by 2/3 when we treat these animals during courtship in order to decrease their sex missing signals. So obviously, the long-term idea would be to try and see if this could work at larger scale, so we can maybe help farmers and be a bit useful and decrease the damages that these flies do in the crops. So that's it. I hope I've shown you that there is the planet Earth is full of vibrating animals, and there is it's very important to study those substrate-bound signaling animals, not just for neuroetological studies and to understand animal behavior, but because it can be helpful to understand the sense of touch and could have potential for fighting pests. And I thought as the finishing slide, I would propose to you to follow the little love story of these two guys here and show you what they're doing in the wild. And this is a cartoon I did with a cartoonist for public engagement purposes. And so we're going to follow these two little guys in the wild. So here they are on their way to a very smelly cactus, prickly pear cactus. This is the male. I think he's going to improve his hairstyle a bit. This is the cartoonist insisted about that. And so the male is starting his courtship. But he hasn't done quite enough here, so it's not worked. And now he's doing the wing and the vibration. That is more convincing to her. So this is the happy ending. So this is it. I'd like to thank the students who worked on what I presented, the project I presented today, especially Eleanor, Stuart, and Monica were the main contributors on these projects. as well as our collaborators and our funders. And thank you for your attention.
Well, thank you very much, Caroline. It was. All right. Still an echo. Well, do you understand with the echo? Okay. So we have time for questions in the room and further in online. So does anyone have a question here?
So, thank you for the presentation. I was wondering if you think these vibratory signals primarily evolved as a sexual interaction signal, or do you have evidence that it's a more general biological signal that has been acted by the male to make the female stop? For example, if you apply the same signal on a male, does it stop moving also?
Yeah, this is a very nice question. Lots of things to say about that. In other species, it's believed that it was originally a predator signal, and that the male sort of uses it to make the female stop. In Drosophila, we looked a bit into it. It's difficult to identify potential predator, but we haven't yet looked at the effect it has on males, but in other insects, in other arthropods, where they look, it can have quite an opposite effect on the male, actually. So sometimes it makes males move quicker, go away, or depending, it can have an opposite effect as it has on the female. We don't know yet for Drosophila, but it's a very nice thing, and we would like to look into it. And for the wing, for the serenade, it's been shown that the male listens to its own song to sort of improve it and stuff. So we don't know if that's the case here as well.
Thank you.
Have you quantified between male variation in their ability to perform this vibration? And is there a link between some sort of male quality, or could it be like an honest signal or something like that?
Yeah. So that, again, very nice question. It would be very nice to know if it gives information about the fitness of the male to the female. The only thing we've looked now is the effect of age. And we found that older males seem to accumulate better in a way. But this is very preliminary. But, it would be nice also to look at the effect of size, obviously, because bigger male their signals might be louder, things like that. Yeah, that's again a very nice project that we should do.
Do you know if fresh fruit transmit or conduct the signal better or differently, and could explain perhaps the difference in Suzuki?
Preliminary data, we know that it transfers well as well. I can't tell you if it's better, or it's going to depend on the species that's on it. I think there might be this aspect of tuning. So you can see that the Drosophila Suzuki signals are very different, and maybe they will be better for fresh fruits. Maybe they'll be more tuned to fresh fruits. Yeah, I don't know yet.
Okay, so we have a question online by Julien Foucault. Julien, when you want, we listen to you. Julien? Okay, so I will just read it. So from the male point of view, what triggers these tremulations? Do you know the mechanism?
This is a very good question. We don't know yet what cues from the female trigger it. We've now we have a paper right now where we look how the male produces this vibration, so which muscles and neurons. If you're interested, it's going to be published soon. But we don't know actually what the female cues, if that's the question, actually trigger them. No.
And we have another question online by Sabina Avozani. Sabina, if you want to ask your question directly, that's possible. Working? No. Okay, I will also read it then. So she wanted to know if the prickly pear cactus is present in the same region of the globe where the species Drosophila melanogaster is thought to be originated.
Yeah, because this is the, I mean, this is not our work, but there's a couple of papers that show that they find Drosophila melanogaster on the prickly pear cactus in the wild, and also the smell of the fruit. The nice, very nice paper by the Benton lab, I think, that showed that the smell was really efficient at triggering male courtship. So the odor of the cactus was a good trigger of male courtship. So this, we don't know very much about fly ecology. We're using it a lot in the lab, but actually there's not much known. So we chose that because there were this couple of papers, but it's not much more known about it.
Are there other questions in the room?
Thank you. So, would females they select against it? Only, so why won't females just select against immobility? Yes.
I couldn't hear very well the question, sorry.
So in Drosophila, there's a lot of mate harm normally.
Male what?
Mate harm. After the mating, there's a lot of harm for the female
yes,
normally.
sure. Yeah.
So why won't females select against immobility, especially because after first mating, I think mate harm. Yes.
Sure. You mean why the female decides to stop?
Yeah, because if it's really costly, the mating is really costly, so normally they should avoid that signal.
But they are, I guess they're convinced to become receptive and to stop by all the male cues, and I guess they are triggered to reproduce. And also, we believe maybe these vibrations sort of pacify the female. Maybe they have a bit of an hypnotic effect, you know. This was interesting. This is a concept we came up with after working with the BBC Natural History Unit. They were saying, oh, is this pacifying the female? And then maybe that's why she stops. And maybe that's the kind of message she receives through her legs are just pacifying her, and then she decides to stop, and with the combination of the other cues. What we would like to know was, and we haven't looked at yet, was how the mated female responds to these signals. It's not clear yet. We're looking into whether they will ignore it, or because we're looking at when we do our experiments, we look at virgin females.
Okay, thank you.
Be a way to use this knowledge against pests?
Well, two ways. One way is that, as I showed you, we've identified some substrates that are like the foam, where you know it's not the very good propagator of the vibration. So you could imagine putting more of this kind of substrate in the crops, right around the crops, and maybe you know the pest pairs are less likely to pour and efficiently and reproduce. And we're trying to develop another way, maybe more active, to try to disturb and blur the signals on the crop fruit as well.
When I can ask a question too. It's a bit of a continuation of the not your question, but the previous one. Because I was wondering what the female uses these signals for, in a sense that it could be so like to get sure it's the right species of male, but it could also be like in the cartoon you had the male like doing his hairstyle, so that's male-male competition for the mate, and so in that sense that would mean that there would be variations among males for the type of signals they would emit, and that would be some would be preferred by some females or others by other females, whatever. But have you any cues of what these are for?
No, and again, that leads to the next aspect, right? Why does the female decide to stop? And we've not looked yet at the central nervous system and see maybe if we identify which area of our brain, are triggered by ultimately by the signals that didn't give us any information on why they should take the decision to stop. It's not clear, but I mean, courtship in general is an association, is a combination of many cues. And usually, I mean, and it's quite accepted that the aim is for a female to agree to recognize that the male is of the same species, probably assess his fitness, and then assess population. But I agree, and sometimes we try to think again about the idea that it's not just going to be a pair of flies like that, right? It's going to be a lot of flies together. So the female is going to have to choose among men that might be more fit. So, no, yeah, that's a very good question. We don't have the answer yet. Like, why do this information trigger her to stop?
Can you reproduce the vibration that the female does and that the male does, and can you expose female to those vibrations or to any other kind of vibration to look at whether it's really specific to what the male does? Or
that's the dream experiment. So for the song, they've been doing it for decades, and still now they are improving the system, and it's acoustics, right? And we've tried, and this is the dream experiment, because then obviously, we could have quantified that, resonant qualitative data. We could put lots of females and then get all of them to stop or not stop, and we could test drosophila stimulants vibration onto just female. But unfortunately, it's been really difficult because basically when you put females in a petri dish, what they tend to do is to stop, basically. And our output here is immobility. So it's been very difficult. We've done some progress with that, but you need to really stimulate the female so that she's by default moving. And so we put pheromones of males in there. We try to. It's been a much more complicated experiment. Setup than we originally thought. But that's the dream. That would be great.
Oh, I have the microphone, so just to continue on this track. Have you tried having several males together, and are you able to isolate the vibrations emitted by each male? So maybe, because if it's for competition between males, you may expect there would be some like a variation, either as a response or as a constitutive variation.
No, we haven't, because it gets very noisy. It's quite impossible to differentiate who is doing what. So no, we haven't. We've tried, but it's just too noisy to analyze. I guess maybe with better software to identify the specific vibration performed by which male, and then to compare it. So it's been tricky.
Do you think that these differences in amplitude between materials can trigger habitat selection in males, for example?
So this again, please?
The difference in amplitude in the different materials in bananas, apples. Do you think that can trigger habitat selection for males?
Yeah, possibly. I mean, it's been shown for the spine bugs, for example, that they prefer. I want to remember the plant. Usually, it's tuned to their favorite feeding substrate, right? And it's been shown, for example, in spine bugs that they prefer to go on the one plant where their favorite food, and actually the signal is tuned for, so it's a much better signal. And that's, if you look in the crops, you'll find them on those plants rather than on other, even though the signals propagate on other plants. But they will preferably find them on those where the signal is the best. So maybe they would choose particular cactus if they could, except in our kitchens, they usually end up around bananas and apples, so they have to deal with it. But yeah, I'm sure, yeah, that would be the case in the wild. Yeah.
All right. No one ever? No one online? Do you want to have a final word?
No, thank you for having me in Montpellier and for all the interesting questions. And I was really happy to see Montpellier. That's an amazing city. So, don't hesitate to contact me if you want.